In this video, we will discuss an experiment in which we will see how phages transfer genes between two bacterial strains. The goal of this experiment is to transfer an antibiotic resistance gene and a gene encoding for a fluorescent protein, GFP, from a donor strain to a recipient strain with the help of phage P1. Our donor strain is called E. coli SAR18. It carries a gene that makes it resistant to the antibiotic ampicillin, and it also expresses the green fluorescent protein, or GFP, which makes it fluorescent. The recipient strain is E. coli M1174, which is resistant to streptomycin, but lacks both the GFP and the ampicillin resistance. By using the phage P1, we want to introduce both the antibiotic resistance and the green fluorescent protein into the recipient strain. In our case, the BLA gene, encoding resistance towards the antibiotic ampicillin, and the GFP gene are in close proximity on the donor chromosome. This makes co-transfection likely. The sequences are located in the so-called ATTB site, where crossover can occur. ATT is short for attachment, and the B means bacterial. The genome of the recipient strain contains streptomycin resistance, making selection possible. In this experiment, we will first prepare the phage lysate from the donor strain E. coli SAR18. Then we will determine the phage titer to assess the concentration of phage particles. Next, we use this lysate to transduce our recipient strain, E. coli M1174. Finally, we will characterize the transductants to confirm successful gene transfer. For the preparation of the phage lysate, dilutions of the phage suspensions are prepared and mixed with a bacterial suspension from our donor strain. After a brief incubation, this mixture is added to a so-called top agar. This agar remains liquid when kept at around 50 degrees. The mixture is quickly poured onto agar plates containing ampicillin, where it rapidly solidifies. Within a few hours, the phage particles infect the bacteria. The addition of calcium and magnesium ions is crucial in this process. These ions facilitate the absorption of the infecting phage particles onto the host bacteria. Without these ions in the culture media, the phage infection process would be much less efficient. In case of successful lysis of the bacteria, plaques appear on the top agar. Plaques are clear zones in the top agar resulting from bacterial cell lysis, when bacteriophages infect and finally kill the bacteria. When phage infection is successful, the so-called confluent lysis can be seen. This means widespread, continuous lysis of bacterial cells. In the first image, the entire bacterial lawn shows lysis, with no distinct plaques. Such plates are used to harvest the phage lysate. Plates with isolated plaques, shown in the other pictures, are not suitable for this. Next, the phage lysate is harvested, and the phage titer is determined. Plates with confluent lysis are selected. The top agar layer is scraped off to collect the phage particles, which are then extracted using a buffer. The resulting lysate is used for transduction of the recipient strain. For this, a dilution series of the lysate is prepared and mixed with a test strain in the presence of calcium and magnesium. This mixture is added to the liquid top agar and spread onto an agar plate. To determine the phage titer, we now need plates with clear and countable plaques. These are selected and counted. Using the volume of the phage suspension and the dilution factor, we can calculate the phage titer. This formula helps us to determine the number of plaque-forming units, or PFU, per milliliter of the original phage suspension. With this information, we can carry out the actual transduction of the recipient strain. A dilution series of the phage lysate is mixed with an aliquot of the recipient strain. To stop the infection process, an excess of sodium citrate is added to the reaction mix. The citrate ions will chelate the cations and prevent further phage infections. This step is crucial to prevent secondary infections and the potential lysis of desired transductants by residual phages. The mixture is then spread on an agar plate containing both ampicillin and streptomycin to apply selection pressure. After overnight incubation, colony-forming units can be observed and counted. With these experiments, we can derive two important parameters. The so-called transduction efficiency shows how effectively the phage has transferred the desired gene into the bacterial cells. It is expressed as the number of transductants per plaque-forming unit, or PFU. It can be calculated using the following formula. The second is the multiplicity of infection, or short MOI. This is the ratio of phage particles to bacterial cells. At an MOI of exactly 1, each phage infects one bacterial cell, resulting in single infections. This condition is ideal for achieving the highest yield of transductants. When the MOI is higher than 1, the total number of infections increases. However, so does the frequency of multiple infections per cell. In such cases, there is a higher risk that bacterial cells will be lysed by a second lytic phage. To verify the successful transduction, we can test the growth of our bacterial strains under different antibiotic conditions. On plates without antibiotics, all strains, transductants, donor, and recipient strains are expected to grow. On plates containing only ampicillin, all strains except the recipient strain will grow, because it is the only strain that lacks ampicillin resistance. Conversely, with streptomycin, only the donor strain will not grow, as it lacks the corresponding resistance gene. Finally, when both ampicillin and streptomycin are present, only successful transductants will grow. These are the only strains that carry resistance genes for both antibiotics. Additionally, the presence of the green fluorescent protein can be easily confirmed under UV light, as seen in this picture. Only the donor strain, SAR18, and successful transductants will show fluorescence, while the recipient strain will not fluoresce. Finally, the successful transfer of the GFP gene can be confirmed using a PCR reaction. For this, we can use primers specific to the gene encoding the green fluorescent protein. Only the donor strain, which is used as a positive control, and the transductants will show a clear band as shown in this agarose gel. This clearly confirms the presence of the GFP gene in the transductants.